My name is Zachary, and this is going to be our group's presentation on the metamorphosis of amphibians. Most animals are indirect developers, which contrasts to human life, meaning that these specific species are characteristically different from their adult forms. As the organism develops, the controlled release of hormones engages many of these same processes as embryonic development, resulting in the changes associated with the transition into the adult form. These are dramatic and rapid changes in cell growth, cell differentiation, and cell death, which yield the sexually mature adult form. Such changes are not only biological, but also ecological, as adults may occupy entirely different niches than their larval counterparts. Species which undergo metamorphosis can be classified based on how different the adult and larval forms are. Primary larvae have morphology that is fundamentally distinct from their adult body plans, which an example of these would be sea urchins, which start off as a bilaterally symmetrical plankton, which becomes a radially symmetrical benthic adult. In this case, the larval plan does not serve as a scaffolding for the adult form, but as rather a radially different organism than the adult form. To contrast this, secondary larvae share the same body plans as their adult forms. This is central to the subject of this video, amphibian metamorphosis. While both adult frogs and tadpoles possess unique structures and adaptations, they are both bilaterally symmetrical and share many anatomical structures which are modified during metamorphosis. The mechanisms controlling amphibian metamorphosis were elucidated in the early 1900s when J.F. Gundernach observed that tadpoles fed powdered horse thyroid glands underwent premature metamorphosis as seen in the experiment shown above. Another study by Bennett Allen strengthened this link, showing that tadpoles with missing or dysfunctional thyroid glands fan to undergo metamorphosis, remaining as tadpoles, which is seen in the image above. Further studies showed that the different concentrations of thyroid hormone were associated with different stages of metamorphosis. This relationship between the changes of metamorphosis and increasing thyroid hormone concentrations was known as the threshold model, which is seen in the slide below. While the reality of metamorphosis is more complicated than this, the model remains useful for understanding the broader patterns. The timeline of metamorphosis begins with premetamorphosis. During this phase, the activation of corticotrophin-releasing hormone, or CRH, leads to the production of steroids, most importantly, corticosteroid. This compound has two likely functions. By acting on the frog pituitary, it produces thyroid-stimulating hormone, which initiates thyroid hormone synthesis. It also acts on the promoters and enhancers of the gene responsive to thyroid hormones, leading to increased tissue sensitivity to these hormones. Of the hormones produced by the thyroid, the most important for metamorphosis are thyroxine and triiodothyronine, T4 and T3 respectively. The process begins with secretion of mostly T4, which is carried through the circulatory system to target tissues. Once taken in, T4 is transformed into the more responsive T3 by type 2 deiodinase. These molecules bind to thyroid hormone receptors, promoting the expression of genes involved in metamorphosis. Some tissues, such as the limb rudiments, contain higher than average amounts of both deiodinase 2 and TRs, allowing them to respond earlier than other tissues. This ensures the functionality of key limbs and systems. Towards the end of metamorphosis, type 3 deiodinase converts T3 into deiodothyronine, T2, which is unable to interact with the hormone receptors. Within the nucleus, the main receptors are TR alpha and TR beta. TR alpha is the most widely spread and is present before the development of the thyroid gland. During the larval phase, the production of this protein increases in preparation for metamorphosis, making it the primary expression factor. This is in contrast to the TR beta form, which is present in initially very low concentrations and activates a variety of genes, including one encoding more of itself. This leads to an instance of positive feedback in which the activation of TR beta during metamorphosis leads to the production of further TR beta, which overall accelerates the response of metamorphosis. Prior to the production of TR3, TR alpha forms a dimer with RXR, a retinoid receptor. This complex associates with gene promoter and enhancer regions regardless of being bound or unbound to T3. However, this state determines how it interacts with the nucleosomes incorporating these genes. When unbound, the TR-RXR dimer acts as a repressor, working alongside co-repressor proteins, notably histone deactylase, to stabilize repressive nucleosomes and prevent gene expression. The binding of T3 to the complex changes its function from a receptor to an activator, recruiting compounds such as histone acetyltransferases instead. This causes the nucleosomes to become relaxed, enabling transfiction and facilitating metamorphosis. Due to regionally specific factors present in the tissues, the exact responses generated during metamorphosis will vary. In some areas, the tissues experience the growth and development of new structures, while the most striking developments would be the limbs of terrestrial adults. A more subtle but equally important development is in the movement of the eyes from the lateral position in tadpoles towards the front of the head. This enables binocular vision, a key trait in the predatory adult lifestyle. Other areas of the body will undergo cell death upon exposure to T3 factors. The obvious examples include the gills and the tail of the tadpole form, which are not present in the adult frog. This transition is achieved through a combination of programmed cell death and consumption from immune cells. In the tail, the initial phase of reabsorption is accomplished via apoptosis of the tail muscle cells, accelerated due to the lack of bone in the structure. Once finished, macrophages digest the remaining debris in the area, and the remaining tail epidermis is shed along with the rest of the larval skin during metamorphic climax. Internally, the red blood cell composition also changes, producing adult erythrocytes, which are better suited to take oxygen from the air. As adult cells are produced, larval cells are sequestered and digested by macrophages in the liver and spleen, respectively. Rather than being generated or destroyed, some structures are instead modified from existing cells and tissues. The intestines of the adult are an example of such reconstructing, as the long coiling tadpole intestines are shortened to better suit the adult's carnivorous lifestyle. This begins with the extracellular matrix of the intestinal epithelial cells dissolving, leading to most of them dying. The remaining cells then undergo dedifferentiation to become stem cells, from which the rest of the intestines develop. Another example can be seen in the development of the adult skull, transitioning from mostly cartilage to mostly bone. For instance, the mandible is derived from Meckel's cartilage, which elongates as bone grows around it. The cartilage structures supporting larval features such as the pharyngeal arch are degenerated, while others are retained in changed form. Interestingly, some tissues are unaffected by the expression of T3. These tissues, such as those of the dorsal retina, express high amounts of deiodinase 3, which works to degrade T3 into its inactive form and prevent the generation of a response. During metamorphosis, the frog's brain also changes. This involves the expression of deiodinase 2 in the cells of the pituitary gland producing thyrotropin, a key part of thyroid hormone expression. The resulting surge of T3 activates genes which block thyrotropin expression, lowering thyroid hormone production and producing a negative feedback loop. This sequence of events is not the same for all amphibians, however. Some genera, such as the axolotl, experience a phenomenon known as neoteny, in which the reproductive system achieves maturation without the rest of the animal undergoing metamorphosis. This may be conditional, such as that seen in the tiger salamander larva in dry environments, or a permanent lack of response, such as the species of the genus Siren. In contrast, amphibians that undergo progenesis metamorphose at a normal rate but mature faster sexually. This is thought to help with the exploitation of new niches in which the immature form may be better adapted. An example can be seen in Bolitoglossa occidentalis, a tropical salamander that lives in trees, benefiting from the juvenile form's webbed feet and small size. Finally, some frog species, such as the coqui frog, are examples of direct development amphibians. Instead of transitioning between a larval and adult form, the frogs develop directly into their adult forms during embryonic growth and emerge as small frogs.